His research interests lie in understanding the pharmacogenetic basis of the immune response to proteins used in therapeutic interventions, as these affect efficacy and safety. His laboratory exploits a combination of computational, in vitro, and ex vivo approaches to understand why some individuals and/or subpopulations develop immune responses while others do not. Work from his laboratory has been published in high-impact journals like Nature Biotechnology, Nature Medicine, Science, and Science Translational Medicine, and Nature Reviews Genetics. He received his PhD from Pune University in India, with subsequent training at the National Cancer Institute in Bethesda, US.
Greetings to all of you attending this conference, and many thanks to Jeremy Fry and the team at ProImmune for the invitation to speak today. Let me begin with something that appeared in the Technologies to Watch feature of Nature in 2019. Last year, researchers reported fresh obstacles hindering the introduction of CRISPR genome editing into clinics. These include activation of the p53 gene, which is linked to cancer risk, unexpected off-target effects such as large deletions, and the immunogenicity against CRISPR systems. If immunogenicity against CRISPR systems were indeed to stymie this immensely promising technology, it would be a tragedy. And thus, I would like to talk today about how to understand and navigate immune responses to CRISPR-associated nuclease Cas9. At the onset, I have no relevant financial relationships to disclose. Before I begin, please allow me a few minutes to thank the people in my lab who've done much of the work I will describe today. Vijay Simadhuri, Wojciech Jankowski, Daniel Lagasse, Lewis Hopkins, Joseph McGill, and Nancy Hernandez. Now, all biologics can be perceived as foreign and elicit immune responses. When anti-drug antibodies develop, these may or may not affect the activity of the drug. Antibodies that affect drug activity by binding to active protein domains are called neutralizing anti-drug antibodies, or NABs, and non-neutralizing antibodies are not necessarily benign, as they can also affect the PK/PD profile and cause loss of tissue targeting. Most importantly, anti-drug antibodies can also cross-react with endogenous proteins or elicit anaphylactic-like reactions. This slide summarizes my laboratory's approach to immunogenicity, which is a perennial problem during drug development and continues into the clinic. So we start with large clinical data sets, and if these are not available, smaller, well-curated data sets, which also have genetic and clinical markers included. We interrogate these data to generate testable hypotheses, which can be probed using in silico tools as well as in vitro and ex vivo assays. Our hope is to elicit some of the rules governing immunogenicity from this exercise, and then use these to redesign proteins for both optimal activity and minimal immunogenicity, which can again be tested using the same suite of tools that we use to generate the rules for immunogenicity. Now let's discuss immunogenicity in the context of the CRISPR-Cas system. An important research focus of CRISPR-Cas technology and a regulatory or safety concern has been the off-target effects, because these are unpredictable and can lead to lethal consequences. Technological successes in resolving off-target effects are moving CRISPR-Cas-mediated gene editing into the clinic. As in vivo clinical applications expand, immunogenicity is likely to be a key regulatory concern. A, because Cas proteins are of bacterial origin, thus in the high immunogenicity risk category per FDA guidance, and studies have already identified Cas9 specific T cells and Cas9 specific antibodies in human populations. Moreover, Cas9 is derived from common human pathogens, and pre-existing antibodies to Cas9 have been identified in human subjects. Speculations about Cas9 immunogenicity are as old as the technology itself. However, it is only since about late 2018 that we have obtained evidence for T and B cell responses to Cas9. The first of these studies showed a high prevalence of reactive T cells in 46 out of 48 donors. The second study looked at both T cell responses as well as pre-existing antibodies to Cas9, and again, a large fraction of the population studied exhibited these responses. The third study from our lab sought to establish screening and specificity assays for Cas9 using an ELISA system, and in a large cohort of about 200 donors, we found that a significant fraction of these individuals carried pre-existing antibodies to both SA and SP Cas9. Based on these studies, we set off to identify potential promiscuous T cell epitopes on Cas9. Our first task was to select an appropriate cohort of donors. We wanted a cohort of donors where the relative frequencies of different HLA alleles were comparable to those in the North American population. And we did this by developing an in-house algorithm. As the graph on the left shows, the donors selected using our algorithm is much superior to that of 10 million randomly picked cohorts of 50 donors each. And we make this comparison using a statistical measure called a Jensen-Shannon distance score, showing that our cohort is much closer to the North American population than randomly picked individuals. Also on the right, you can see that the relative frequencies of different HLA alleles is very comparable in our cohort and that in our population of interest, again, the North American population. In addition to the cohort of donors from which we obtained the PBMCs, we also generated 209 15-mer peptides that span the entire sequence of Cas9. We pooled these peptides, generating 21 pools of peptides in all. To look at T cell responses, we selected 3 cytokine markers and 2 co-stimulatory molecules to have a more comprehensive sense of all the responses to Cas9. Moreover, we chose to take a flow cytometry-based approach so that we could look at these responses separately in CD8 positive and CD4 positive cells. Here are some results. So what you see on the x-axis are the different peptide pools, and on the y-axis we have the number of responders, that is the number of donors who responded with respect to each of the markers which are shown in different colored graphs. Again, we plot this data separately for CD4 positive cells and CD8 positive cells. Now, which donor is a responder? To do this, we determined the percent of cells in the relative quadrant from the unstimulated sample and used that as a background frequency. A one-sided binomial test was used to decide if the response was significantly greater than the background. In determining significance, the p-values were adjusted p-values, and we accepted a false discovery rate of 0.1. Now, these data, while comprehensive, are difficult to interpret. So what we did was enforce a rule wherein a positive response for any combination of 3 or more of the 5 markers was considered as an appropriate measure of T cell activation. Now the simplified graph is shown over here, and you can clearly see the number of responders for each of the peptide pools. And you can again, look at the data separately for CD4 and CD8 positive cells. All the analyses shown previously are based on the criterion of whether responses were significantly greater than baseline. However, as the heat map on the left shows you, even among positive individuals, there is considerable diversity in terms of the intensity of the T cell response. Now, currently, the association between stimulation of T cell activation markers and clinical immunogenicity is poorly understood. developing algorithms for immunogenicity risk assessment will need more clinical data, and more comprehensive algorithms will likely incorporate some combination of the breadth, that is the number of T cell activation markers responding, and the depth, that is the stimulation index for each activation marker of the response. Most protein therapeutics are delivered directly into the bloodstream as the intact protein. However, Cas9 immunogenicity and its assessment is likely to be far more complex because Cas9 may be introduced in vivo as DNA, as mRNA, or as ribonucleoprotein particles. Thus, the delivery by Cas9 DNA or mRNA would engage MHC class I molecules and CD8 positive T cells, whereas RNPs would most likely engage the MHC class II and CD4 positive T cells. Immunogenicity data thus should be assessed considering the role of MHC restriction in T cell activation, and in our MHC-based assessments, the appropriate pair of T cell subpopulation and MHC class are used. Here, we have tried to estimate which MHC class 1 and class 2 alleles engage with each of the Cas9 peptides. So if a MHC allele and a peptide are highlighted, that means that that particular peptide elicited a response in all donors with that MHC allele. Again, we have analyzed this data with respect to CD4 positive cells in determining the MHC class I responses and CD8 positive cells in relation to MHC class II alleles. The lower graphs are promiscuity scores, which are the number of MHC alleles that have been engaged, or the fraction of MHC alleles that are engaged, weighted for the frequency that they occur in the North American population. These data, while interesting, have one significant drawback. By directly using peptides, we did not gain an understanding of Cas9 protein processing and presentation in antigen-presenting cells. Investigations using MHC-associated peptide proteomics could better elucidate which protein regions are most relevant in predicting patient risk. Here is some MAPS data. On the y-axis, you see individual donors, and the x-axis represents the different positions on the assay Cas9. Each of the small squiggles represents a peptide that was identified. Note that at any single location, the length of these small dashes varies, illustrating that peptides of different lengths were identified at each position. There are clearly some areas on the Cas9 represented by blank columns, which essentially illustrate areas where no peptides were identified from the Cas9 protein. In this plot, we illustrate the binding affinity of each of the peptides that were eluted from MHC alleles. So each individual plot represents a particular donor, and we are measuring the binding affinity of the eluted peptides for the HLA alleles in that donor. And you see that they all have, the gray areas predominantly have high affinity peptides, or at least the graph is skewed in terms of high affinity peptides. For context, the blue dotted line shows that peptides with all percentile ranks occur at relatively equal frequencies if you consider all the peptides in the Cas9 protein. To make the data more useful, we have overlaid the T cell proliferation data obtained using individual peptides on the MAPS assay obtained using the full-length protein. Finally, to hone in on T cell epitopes that are biologically relevant, we identified peptides that are presented by antigen-presenting cells and also test positive in a T cell stimulation assay. And at the end of the day, we are left with just 9 peptides that appear to be really high risk in the Cas9 protein. To summarize, given the large size of Cas9 and poor homology to human proteins, modulating Cas9 immunogenicity appears at first blush to be a daunting task. The maps used in conjunction with T cell proliferation assay suggest that the problem, while challenging, may be more manageable. It is, however, important that the assays and analytical tools described here are validated and fit for purpose, as they will be central to clinical decision making, such as, for example, which patient enters a clinical trial, which patient is at very high risk of developing immune responses, etc. Cas9 immunogenicity is clearly a very nascent field, and there are some clear unmet needs. For example, we need assays that are validated, reproducible, have defined cut points, and are fit for purpose. For instance, if studies do not have consistent and validated methods and statistical tools for determining a cut point for identifying a subject as positive, there will be wide variation in the results. The model systems need to have meaningful clinical association and be representative of the population of interest. For instance, cells for ex vivo assays should be sourced from a cohort of donors that is representative, that, for example, vis-a-vis HLA distribution of the population that will actually receive the treatment. It's also important that the reagents are well characterized, preferably of GMP quality, and if not, the contaminants and impurities should be carefully controlled. For instance, academic researchers procure laboratory-grade Cas9 from numerous suppliers. These are varying amounts of endotoxin which affect responses in assays used to measure T cell responses. For instance, when we looked at endotoxin levels in 7 commercial off-the-shelf Cas9 reagents, we found that the endotoxin levels varied by several orders of magnitude. Similarly, these products gave very different interferon gamma signals in an ELISpot assay, even when we used the same donors. Interestingly, when we depleted endotoxin levels in 4 of these products, we found that the interferon gamma signals were all comparable and at the same baseline. I would like to conclude with a small sampling of headlines about Cas9 immunogenicity. I would argue that it is not the insurmountable technological roadblock that it is made out to be, but it is a challenging proposition. We do have, however, the tools, resources, and reagents to surmount this eminently challenging problem. I would like to thank the audience for their attention, the organizers for their invitation, and I'm happy to take any questions.
Great, thank you very much, Zuben. If you'd like to unmute, do we have any questions for Zuben? Hands up, you can raise your hand or you can put it into the chat as well. So, Zuben, I have a quick question for you, just in relation to the plot where you were talking about with the prediction of the affinities of those peptides that were identified in the MAPS assay, with the dotted line along the baseline and then the high affinity peptides that were shown. You mentioned that that was for each of the different donors. Was that for all of the HLA alleles that are expressed in that donor, or was it just looking at DR? I'm trying to remember exactly.
So we looked at the all the DRB alleles, because these are the most commonly associated with clinical responses, And yeah, so essentially this is just a kind of, it's, I wouldn't call it really a quality control, but it is an expectation that you have from these results. In most instances, the peptides you pick up would be the high affinity ones. And that's what, we just check these. And in some instances, you do find that they are, in a couple of donors, they tend to be towards the right of the curve where you won't really expect them to be, but, so it's
Yeah,
a way of,
no, thank you.
of revisualizing the data.
Yeah, yeah. Okay, and I think we've got a question here as well from Hylam. So Hylam asks, do you think we should look into the Tregs among those CD4 populations? Hylam, if you want to unmute, you can also ask any clarifying points if you want to.
Yes, so there are, I mean, given the severely limited amount of actual data we have about Cas9 immunogenicity, there is some indication that there may be some regulatory effects in addition to the activating effects. So yes, I mean, as far as Cas9 immunogenicity is concerned, it's a wide open field as far as research is concerned, because frankly, what I see is there is far more speculation, commentary, etc., and very limited amount of data. And as I mentioned in my talk, this is a very different way of using it is not really the therapeutic, but you have therapeutic outcomes, and the delivery methods, everything is very different from our experience with protein therapeutics. So, I would like to see data on all aspects of Cas9 immunogenicity. And I mean, there is, normally, I generally argue that, because all animals will elicit a response to a human, or in this case, a pathogenic protein. So animal studies are not very useful when establishing immunogenicity risk for human proteins. But in this case, because we absolutely have the information is so limited, that I think animal studies, at least at a basic science level, would be very useful to have at this point. Of course, using all the attendant technologies that we have available to us.
Thank you. Any more questions? I have another sneaky one if nobody else does. I want to, I'll take advantage of the fact I've got you here. So one of the things I was just thinking about was, I know you were obviously emphasizing the importance of obviously looking at the treatment patients, sorry, the patients that were actually going to be treated with this in regards to obviously understanding the immune response. It's kind of really important to do that. And obviously the genetics are quite different in different ethnic groups. Having said that, I guess it was quite interesting from the MAPS data, overlaying that with the T cell proliferation data, that there were some very, there was a relatively limited subset of sequences that were kind of coming up. Do you think, and it's a bit of a, an unknown, of course, but if we were to look at a much wider population, looking at sort of Indian population, Japanese population, Chinese population, etc., then actually, are we still? There's still going to be a kind of a limit to what we need to kind of think about in regards to any reduction of immunogenicity to Cas9.
Yeah, that's an interesting question, and I have thought about it. And so, I mean, it could work out in one of two ways. There may be limited number of peptides of interest globally, or there may be multiple limited subsets of peptides for and either way, I mean, it would be important information to have in terms of generally for somebody who's at not too, it won't be too much in the future, somebody planning to develop a strategy for using CRISPR-Cas9 for in vivo treatment, that whether there would be what is their actual population pool in terms of. But at the end of the day, and you mentioned it in your talk as well, that as costs come down, these assays are better validated. And given the, I'm assuming that when these treatments hit the market, there will be stupendously expensive. So there is probably a space for, carrying out these assays for an individual before, so I mean, we don't need to, guess about, what it might be in terms of the population. Just, go ahead and do at least some of these assays for that individual before treatment begins.
Yeah, great. Well, thank you very much indeed, Zuben. Appreciate you joining us today. And thank you again for your time.
Thanks.